Welcome to the series in food microbiology. In this series, in the last couple of lectures, I've been dealing with how to determine that a given food sample has microbes present in it. What are the different detection methodologies? We are continuing with the same type, how we are going to deal. Today we are going to talk about the indirect and qualitative estimation of microbial content from food samples. In the earlier lectures, we saw the sampling strategies, meaning from if you are given a lot of food, from among that lot, randomly how will you choose the sample, and then what were the attributes, like did you keep a marginal level that above this threshold level you are going to have accept that the food has microbial contamination, and below that whether you are going to whether it can be accepted. So, the different types of sampling strategies as well as some kinds of detection methodologies we saw in the early lectures. In this session, we are going to see the indirect and the qualitative tests for microbial enumeration. The first among that is dilution to extinction in non-selective broths. So, this method, like for example, if you take a food sample, then take a swab, you can wash the sample and collect the wash, and then this wash is serially diluted and transferred to an aliquot. For example, when you serially dilute it, we dilute like 1 to 0.1 ml, and finally, so what you do is you have a series of tubes in which you take the saline or the broth, and you have the first wash which you have collected from the food sample. So, that would be the master sample. From that master sample, say for example, I am adding 200 microliter or rather 0.2 ml. Okay, I take 0.2 ml from the sample from the wash, add it to the first tube. So, that is diluted. Now, mix this well, and from this tube I take another 0.2 ml and add it to the second tube. So, serially we will keep on diluting it. So, as it comes to the 10th tube or the 12th tube, the microbial volume would actually become very dilute. So, this would be reflected on the rate of growth when you keep the tube for incubation at optimum temperature, and after a couple of hours when you observe it, you will see this difference in the rate of growth in the turbidity present in the tubes. So, this is followed by incubating the tubes at a specific temperature for a specific period of time. So, when the tubes are examined for the presence or absence of growth marked by the turbidity of the broth, with the highest sample of dilution that gives the growth, and assuming that this tube has 1 to 9 viable cells of the group of interest, then we can count the microbial cells per millimeter or gram of the sample. We can estimate, there is a formula for making the calculation for that. The most probable number or the MPN in a selective broth. In this method, aliquots from a serially diluted sample are inoculated in a broth in tubes, having one or more selective agents that facilitate the growth of the selected microbial groups present in the food. So, what is the difference between the previous method and this method is that here when you are doing the dilution, the medium where you are doing the dilution, that contains a selective agent which in turn will make the microbe proliferate fast, and all the other microbes would be either inhibited or become dead. So, you are selectively making a particular microbe to grow in the medium. So, if your food sample was contaminated with that microbe, now this becomes very important, especially when you are talking about a pathogen, because we already saw that the pathogens are usually less in number when compared to the normal microbiota, the flora which is present on a food sample. So, you are selectively asking those particular microbes which can grow in the presence of selective agent, they would grow in the medium in the serial dilution, whereas the others would be inhibited. Generally, 3 to 5 broth tubes in each dilution and a minimum of 3 consecutive dilutions we use. So, after incubating at recommended temperature and time, the broth tubes in each dilution are scored for the presence and absence of the growth. So, after doing this, you keep it for incubation at a specific temperature and for a specific time, then take out the tubes and observe whether then you score for yes growth or no growth depending upon the turbidity level which is seen in the tube. From the number of tubes showing the growth in each of the 3 successive dilutions, the number of viable cells of the specific microbial group can be estimated from the available statistically calculated tables, and the method gives a wide variation. So, from the observation, visual observation, now based with the statistical calculation which is available, you can make out how much the microbe has grown, what is the rate, what would be the amount probably present in the original sample concerned. MPN methods are quite often used to estimate coliforms and fecal coliforms in food and water by using brilliant green lactose bile broth, otherwise called as, this is called as a BGLB. B for starting for brilliant, green, lactose bile, BGLB, broth and easy broth. So, if these two specifically will allow the coliforms and the fecal coliforms to grow, at the same time they will inhibit the other microbes. So, the color change here would indicate that the microbes have grown, the presence or absence of the microbes concerned. The dry film and related methods. A rehydratable dry film method consisting of 2 plastic films attached together on one side and coated with culture media ingredients and cold water soluble gelling agent. This was developed by 3M company and this was called as a Petri film. Okay, so, you have 2 plastic films which are attached together, and onto this plastic film you are going to coat the culture medium with cold water soluble gelling. So, you are totally avoiding the hot agar stuff, and you have a cold water gelling agent. So, this method can be used with non-selective ingredients to make an aerobic plate count APC, and with selective ingredients certain specific groups can be detected. So, you can make it as a general microbial growth structure with a aerobic plate count where any aerobic organism which is present on the food sample would grow, or if you add a selective agent to the particular culture medium, then this can be used to enumerate the specific microbial group we are talking about. Ready gel. This is a plating medium that does not use agar as a solidifying agent, and this is employed by inoculating a re-sterilized ingredient with food homogenizers or diluents, followed by mixing and holding to allow for solidification, which occurs in about 30 minutes. So, here in ready gel you do not have any agar, but instead what we do is the re-sterilized ingredients along with the food homogenizers, they are mixed up together and then allowed to solidify. We mix all the ingredients and allow them to solidify, which will happen in about 30 minutes. This is normally used for enumerating cyclotrophic organisms because there is no exposure to hot molten agar, which can lower the numbers of cyclotrophs since some are extremely heat sensitive. Now, when we are talking about checking microbial count of refrigerated products, for example, meat which has been kept for refrigeration, which has been kept frozen and it has been already kept for some time. So, after a particular number of days, you now want to take it out and check whether there is microbial growth there. Some of the cyclotrophs are extremely sensitive to heat. So, the moment you use a pour plate in this with molten agar, hot molten agar, they will die. Then you will see a blank plate, you will say no microbe, but that would be wrong because the extremely heat sensitive microbes actually will die in the presence of molten agar. So, in such a condition, to allow the growth of such cyclotrophic organisms, we can use this ready gel, and the colonies on ready gel tend to be very small in size. On the basis of this, the company has developed another 3M ready gel orange serum test. This is actually very useful for the juice industry to test whether the juice, either the fresh juice or the refrigerated juice, has microbial contents, and this is used to detect acid tolerant putrefactive microbes without differentiation of those microbial types. So, what we are trying to do is we are going to just detect whether there is a microbe. We are not going to say yes, this is this microbe, this is this microbe. Such a distinction is not going to be made. We are going to just check whether there is a microbial growth or no microbe present in the frozen juice or the sample juice. The low pH of the medium inhibits the growth of the other bacteria. So, only acid tolerant, for example, orange juice, it is acidic. So, normally itself only those organisms which can sustain and reproduce in the acidic pH would be growing there. and they are the ones which are going to cause the spoilage to this juice concerned. All the other organisms would have automatically dead because of the pH of the medium concerned. So, this is highly sensitive, as the test format can accommodate a 1 ml direct sample of the product onto the orange serum medium. So, you take the medium and then add 1 ml of the juice, that would be enough. So, director you are trying to find out whether the given sample has microbe or not. The next methodology we are going to talk about is called as a simplate. This is a culture method that is based on the activity of several enzymes common to many food organisms. So, we are not going to distinguish a particular microbe, but what we are going to see is you are going to see a broad range of organisms, detect a broad range of microorganisms which are normal dwellers. These would be spoilage, not specific pathogens, the spoilage organisms, which is a heterogeneous mass, not a homogeneous mass. And we are going to take enzyme activity as the mark. So, many of the metabolic pathways would be common among bacteria, and these metabolic pathways are catalyzed by enzymes. So, based on this enzyme activity, we are going to detect that yes, this type of microorganism which use these enzymes are present in the particular food sample given. The growth medium contains substrates that are hydrolyzed by enzymes to release MUG. This is methyl umbelliferyl glucuronide, and this is a fluorescent compound, and this fluorescent compound is visible under long wave ultraviolet light. The special plates have holes or wells, and they come in two sizes, 84 or 198 incubation wells. So, this is the plate which is seen with small holes which are visible here, and this technique is in essence an NPM method. Unless the conventional plating methods, this does not allow for characterization of the colony features. So, here if the sample contains microbes, so and those substrates, so you are going to keep it on the medium which contains substrates which can be hydrolyzed by the microbial enzymes releasing MUG. Once MUG is released, it is fluorescent, and that can be detected by the long wave ultraviolet light. Dye reduction tests. This method is based on the principle that some dyes like methylene blue and rizazurin are colored in oxidized states, but colorless under reduced conditions, and this change from oxidation to reduced condition is because of microbial metabolism and growth. So, here we are going to use the oxidation reduction potential, whether the dye is occurring in an oxidized state or whether it gets reduced because of the growth of microbe. That would be reflected as a color change, which indicates yes, if microbes are present or no, microbes are not present. Properly prepared supernatants of foods are added to standard solutions of either dye for reduction from blue to white for methylene blue and from slate blue to pink or white for rizazurin. So, we take out the sample, a swab or a wash out from this food sample, and this is added straight to the dye. Okay. And once, depending upon whether we are going to use methylene blue or whether we are going to use rizazurin, if it is methylene blue, if the wash sample contains microbes, then you will see the change in color from blue to white as the microbe grows, or if it is from rizazurin, from slate blue to pink or white in color depending upon the microbial growth. This will be a gradual change depending upon how the microbes grow, the rate of microbial growth. So, the moment you add the wash, you are not going to see the color change, but if the food sample contains microbes and then you incubate the sample plus dye mixture under optimum conditions, as the microbes start multiplying, replicating, and the enzyme activity starts, you will see the gradual change in the color. The time for dye reduction to occur is inversely proportional to the number of organisms in the sample. Because microbial groups in a mixed culture in a food differ in the rate of metabolism and growth and the ability to reduce the environment, this method is not considered very accurate and effective with different foods. Now, the drawback of this is you get a heterogeneous microbial load from a food sample. And in the heterogeneous microbial load, each microorganism have a different rate of metabolism and different rate of growth. For example, all of you know that E. coli has a generation time of 20 minutes, which means that one cell will divide into 2 in 20 minutes, then 4. So, like that every 20 minutes you are going to, which means it is very rapid, whereas yeast and some other cells will take 24 hours or 48 hours to divide. So, and accordingly, so depending upon the growth rate, depending upon the rate of metabolism, this change will differ. So, the reduction time actually will vary, especially we are going to talk about a heterogeneous mass of microorganisms which have been taken from the food sample. So, this methodology would not be, it will tell you that microbes are present, but it would not be very accurate for predicting that these are the microbes which are present. But you can assess the microbial quality of the food based on the color change, and this method is generally used to determine the microbial quality of the raw milk. So, here we have added methylene blue to milk sample. You see the blue color here, and when you keep this tube for incubation at 37 for some time, say, and you look for the color change, gradually from this deep blue color, as the microbial rate increases, growth rate and metabolism increases, this will get slowly, the intensity of the blue color will reduce, and at the end when the microbial rate becomes completely, it will change into white. So, this indicates the rate of microbial growth, number 1, and 2, that the raw milk which was tested does contain microbes. So, the efficacy of pasteurization can also be detected using this, whether there has been spoilage or whether there is a contamination of raw milk with pasteurized milk, that can be detected using this methylene blue reduction test. The other common methodology which is used is using triphenyl tetrazolium salts and the derivatives. These are normally colorless, initially colorless, but as microbial growth happens, as time proceeds, they become very strongly colored, and usually they are insoluble. After reduction, they form formazans. Triphenyl tetrazolium chloride itself is the most widely used component of diagnostic and selective agar media, on which some bacterial colonies will become dark red to maroon as formazan becomes precipitated within the colony. So, when you use triphenyl tetrazolium chloride as a selective agent in the agar plate, and you plate the bacterium on it, depending upon whether the bacterium is able to utilize it and then reduce it to formazan. When the dye gets reduced, it becomes intense red. So, slowly the colony will take up the red color, dark red to maroon, the deep maroon color would be visible in the bacterial colony because of the accumulation of the formazan. So, this will indicate that microorganisms are produced, and they are capable of utilizing tetrazolium salts and then reducing it into the formazan present. The crystals of formazan produced from INT are so intensely colored that they are readily seen in individual microbial cells under the microscope, and their presence may be used to assess the viability of the cells. So, here viable cells. So, suppose you have a mixture of microbes where some of the cells are dead and some are alive. So, normally you won't be able to make out how many cells are dead and how many cells are viable or living. So, but in this case only the living cells will do the reduction, and they will show the red color. The dead cells will not be able to take up. So, the number of viable cells which are present also can be detected using this particular dye. Qualitative methods to isolate microorganisms. The first step here now becomes very important, because we are going to look for pathogens. We are not looking for the heterogeneous non-pathogenic spoilage organisms, but we are going to find out whether the given food contains any lethal organisms which can cause intoxication, food intoxication, mild food poisoning to lethality, for example, your Clostridium botulinus toxin. So, you need to find out whether the pathogen is present in the food, and we already saw that usually pathogens would be very less in number when compared to the general microbiota which is present in the foodstuff. So, the main objective of this method is to determine whether the sample contains a viable cell or spore of a specific pathogen. Not only a viable cell, but spore. In the case of spore formers, whether the spore of the organism is present. Food tested for several pathogens such as Salmonella, E. coli 0157:H7, Listeria monocytogenesis, Vibrio cholerae and Shigella by specific isolation procedure when necessary. So, these Salmonella, E. coli 0157, Listeria, Vibrio, Shigella are the common pathogens associated with various foodstuff which cause extreme cases of food poisoning. So, even if these organisms are present in very less number, detection of this becomes very important, number one, and especially in ready to eat products, and if you are going to market or export these foodstuff, the presence or absence of these, you have to give a certification saying that the food is safe. So, then for that randomly you will need to test the samples for these. So, testing for the presence or absence of these foodborne pathogens becomes very important under such conditions. Other than those, the other enteropathogenic organisms of E. coli, Yersinia enterocolitica, Campylobacter jejuni, etc. For here we do not use isolation procedures, but for these three normally we use enumeration, counting. But for the case of the previous extremely serious pathogens like Listeria, etc., we need to know not only enumeration, but even if it is present in very less number, we need to isolate them. An isolation procedure generally contains several steps, such as the non-selective pre-enrichment, followed by selective enrichment, then testing on an agar medium containing selective and differential agents. So, the procedure is like, first, since the pathogens are going to be very less number, initially you will select all the microbes which are present. So, you take a wash or you take a swab, okay, and you add it to the medium, and then in that particular medium which is non-selective, it is not going to select this microbe or that microbe, but if microbes are present, that heterogeneous pool will be allowed to replicate in that particular medium. So, it is a non-selective pre-enrichment. Then from that non-selective pre-enriched medium, you are going to go for selective enrichment. So, in this selective enrichment medium, only specific microbial groups, if present, would be growing, all the others would be inhibited. And those microbes which grow in the selective enrichment medium, those we test on an agar medium containing selective and differential agents. So, maybe with using bacterial antibiotics which will inhibit a particular thing, or you use a biochemical markers which will indicate, or maybe a detection by the presence of gas detection. We can use a dye detection test where the color of the colony will change, or the area around where the colony grows would have a change in color, indicating that it is this particular organism which is growing. So, using an agar medium containing selective and differential agents, we can detect that yes, this particular pathogen is present in the particular food sample concerned. It is assumed here that the food normally contains a low population of the pathogen as compared to the associated microorganisms, and the pathogens could be in an injured state. Not only is the pathogen going to be less in number, but it can also be injured during the processing. So, we have to allow it the recovery time, get normal, then proliferate, then selectively enrich, and then isolate that. The food sample, for example 25 grams, is first pre-enriched in the non-selective broth and then incubated for the injured cells to repair and then multiply in order to reach moderatively high numbers along with many other microorganisms. So, this incubation period in the non-selective broth will give time for the pathogen which is already less in number and which might be injured. So, it will now have a conducive medium to repair itself, start its metabolic activity, then start slowly multiplying. So, here after that, once it has repaired itself, it will start reproducing faster. So, it will reach into a higher number, moderately high number. So, now this higher number, when you transfer to the enrichment broth, selective enrichment broth and then incubator, automatically the other microbes will be inhibited. It is only going to be your pathogen which is going to be multiplying at a faster rate. And during incubation, the specific pathogen and closely related microorganisms will selectively grow to high number. Many of the associated microorganisms will not grow. A small amount, usually at the rate of 0.01 ml of the enrichment broth, is then streaked onto the surface of a pre-poured selective differential agar medium plate, which is then incubated for a specific time for the colonies to develop. So, once it has grown in the broth, you take an aliquot from the broth and plate it onto the agar plate, which also contains a selective differential agar. So, what grows up there is going to be only your pathogen. And from the presence of the differential colony characteristics, the specific pathogen can be tentatively established. So, we are saying that because the colony grew up in a selective and differential medium where only these genera or this particular can grow, tentatively the food sample contains this particular pathogen. But a tentative identification is not enough, you now need to confirm this. So, this tentative identification, we call it as a presumptive test, and this presumptive test has to be confirmed. For confirmation, the cells from the characteristic colonies are purified and examined by recommended methods for biochemical reaction profiles and serological reaction against a specific antibody. So, now what we do is, from the confirmed cells of the presumptive test, we will look for a specific biochemical reaction, which can be diet reduction, it can be a color change, or it can be the production of gas, or it can be because of a fermenting biochemical reaction. Any of these will confirm that yes, this pathogen is present. Or the other option is you have already raised an antibody against the flagellum or a cell wall component of the particular pathogen. You use those antibody. So, that antibody, because it is against that particular pathogen, it will go bind and give a fluorescence or some reaction which you would be able to judge, saying that yes, the food sample contains this particular pathogen, we have isolated this, it is confirmed that it is this particular microbe which has grown. So, based on this, so we have isolated a pathogen. Now, the basic test, which is water testing for the potability of water. This is actually very important, because not only the normal drinking water, but we are also going to look for the water which we are going to use for processing of food, then water which you are going to use for washing, cleaning, any food sample, or that has also to be microbial free. It cannot be microbial free, but at least it should have the threshold level which is accepted. It should not be beyond what we call as a threshold level. So, the potability of water. For this, normally we use what we call as a coliform test technique, or it is otherwise called as a multiple tube fermentation test. This is a standard method followed all over the world to determine whether water is potable or it is fecally polluted. And the technique involves 3 successive steps: the presumptive test, the confirmed test, and the completed test. In the presumptive test, lactose broth tubes are inoculated with 3 different water volumes to give an estimate of the most probable number of the coliforms in water. See, so what we are going to do is, you have 3 lactose broth tubes. Into each, you are going to inoculate 3 volumes, different volumes of the same sample, say 1 ml, 1.5 ml, 2 ml. So, now what will happen is that, if the amount of microbe is very less, 1 ml, the lactose broth where you have added 1 ml might not show much of microbial growth, but 2 ml will definitely show, because the microbial load would be much more there. So, the tubes which are positive for gas production are now inoculated into brilliant green lactose bile broth in the confirmed test. So, what happens when you inoculate the tube with water sample, you incubate it, then you see what you call as gas bubbles. So, you isolate those tubes with the gas bubbles, and then those are the samples which are going to be taken and inoculated into the brilliant green lactose bile broth. So, this would be, if you still have a color change here, this is a confirmed test, and then positive tubes are used to calculate the MPN value. The presence of the coliform bacteria in water samples are established in complete. This, for example, you are seeing E. coli growth in the EMB agar plate. In the last lecture also we talked about the dark greenish metallic sheen which confirms the presence of E. coli. So, in this particular lecture we saw how the different dye reduction methods as well as the ready gel or the simplet mechanisms, how we can detect the presence of microorganisms. Then if pathogens are present, then how do we enrich and isolate those pathogens and confirm that yes, the food sample cannot be consumed, it has to be rejected because it contains a food poisoning agent. In the next couple of lectures, we will see the other methods which are going to be used. Now, if you are given a random food sample, how do you think, which would be the steps you would take to find out, 1, whether a microbe is present, 2, identify and enumerate the microbial colony which is present there. Thank you.